It's my distinct pleasure to introduce to you all a new colleague here in the Diabetes Center of Excellence, Dr. Accalia Fu. We call her Callie Fu. I just got back from this big diabetes meeting in LA, and I think a very exciting theme is that all that we've learned and taught about type 1 diabetes being the immune system attacking these poor innocent beta cells has recently at least been amended a bit now that we have increased ability to look at the beta cell itself, because all sorts of efforts that improve beta cell health also seem to influence the immune response against the beta cell. And Dr. Fu has been doing that type of research for a number of years. She grew up in Canada near Ontario, where she received her undergraduate degree and her PhD before moving in 2014 to the Dana-Farber Cancer Institute, where she's done considerable work on beta cell metabolism and its role in interacting with the immune system. So we were thrilled to, during our national search, we were thrilled to recruit her to join our program. And you can see what she'll be talking to you about today, metabolic licensing of beta cell survival and inflammation. So it's my great honor and pleasure to introduce you all to Dr. Fu. Thanks for joining.
Thanks, Dave, for the kind introduction. And the nice introduction to my research as well, which really kind of has dug into the potential behind the ability to license beta cell survival and license metabolism in order to influence beta cell survival and inflammation. And I came to this because of kind of more my broad interests scientifically, which I'll just briefly introduce those ideas to you. And that is kind of overall in metabolism specifically at the molecular level. So all the metabolites floating inside and outside of your cells and their ability to influence a cell's adaptation and in turn help determine cell fate, whether a cell will respond to different stressors to move towards cell survival or self-termination or growth and expansion. And I think it's a really exciting time technologically with the advances that we've made to be studying this question, and also our appreciation for the importance of metabolism in cell adaptation. And this is kind of how I think of metabolism. It's sort of like a roadmap that I've shown you here. And in red, I've highlighted all the pathways in human metabolome that we can measure to date. And in blue is all the pathways in mouse or in rodents. So you can see there's a lot of purple, which is the overlap. But at the same time, you can also see areas where there's just red or just blue. And I put this up here just to make the point that it's, I think, really important that we actually consider this in our studies and not only rely on rodent models, but also try to recapitulate that in human tissues. And that's a real challenge for any of you who work around pancreatic beta cells or islets, as you know. But to the best of our ability, we try to recapitulate any findings in the human system. As I mentioned, I think we've come a long way with the technological advances from actually early and modern biochemistry, which started with the study of metabolites and the chemistry of metabolism, basically. But they were doing so without having the ability to directly measure these things. So I think that was pretty remarkable. And we've come a long way from there. We have this appreciation now that metabolites are not just sitting there as products of reactions, but they're actually able to take part in determining if a cell process will take place or not. So I've highlighted some of those cell processes on this map. And not only do metabolites serve as substrates, but they can also serve as cofactors for enzymes, transcription factors, and translational machinery to really help determine a cell's ability to respond to its environment. It's actually a really fast way with metabolites changing in the environment. It's kind of an immediate way that a cell can change and adjust what it's doing and respond to that. So I think we have a lot of potential if we can tap into this capacity for molecular metabolism to influence cell fate. And I'm particularly interested in the context of inflammation. And I've studied this in the pancreatic beta cell. And I'll take you through some recent work I've done that describes how I've identified a new and kind of unusual link that glucose forms And so I've looked at glucose metabolism in beta cells for a number of important reasons, and I'll get to that in a bit. But I've identified an unusual link it forms to the Urea cycle and a direct link as well to de novo glutathione synthesis. And these two metabolic links actually provide a protective effect in beta cells and help them adapt to inflammation stress. So why do we care about inflammation stress in beta cells? Well, I don't think I have to introduce this idea to this audience, but in actually both forms of diabetes, type 1 and type 2, there's what's referred to as sterile inflammation going on, or autoinflammation when it's in the absence of any overt infection or pathogen. And we know that this contributes to impairing beta cell function to secrete insulin, increases beta cell death, and ultimately leads or contributes to hyperglycemia. And that dysregulated hormone signaling combines with and contributes to metabolic dysfunction in liver, muscle, and fat. And the things that I've noted on this side of the slide in this context actually refer more to the setting of type 2 diabetes, but I just put this up here to mention that the metabolic dysfunction we see, hyperglycemia, dyslipidemia, IR, in these tissues, we've characterized very well at the molecular level how and what signaling pathways could be involved in how this contributes to local tissue inflammation within those tissues. And aside from that, we also have some information, even from more than 20 years ago now, that, for example, fat can release pro-inflammatory cytokines in this context of dyslipidemia that then can go on to directly enhance beta cell inflammation as well. So we have a lot of information on this side of the slide, but surprisingly, we actually know very little in comparison when it comes to the beta cell and how beta cells might be contributing to inflammation. So as Dave mentioned before, that's kind of one of my big motivators right now, is identifying what are the features inside beta cells that are actually making them more immunogenic, perhaps, or helping them be more sensitive to the immune attack. And so in this sense, what I'm first considering is looking at metabolism as one such feature that would be important in how beta cells might be contributing to inflammation. And that's because metabolism is very unique in how beta cells handle different nutrients, which I'll get into in a little bit. But I also first wanted to give some clues from clinical observations that we've made that this is actually a relevant and important question. And so some of those are that we can see, so in the context of type 1, in some cases, we can see siblings of individuals with type 1 that might have one autoantibody, but yet the levels will go up and then down, and they may never develop diabetes. So it's not always 100% used as a diagnostic tool until you get several autoantibodies being produced over a period of time. And then we also see with the main mutations that are responsible for the majority of familial type 1 diabetes in the HLA and MHC regions. The most penetrant ones there are the DR3 and DR4, but people with those mutations really only have about a 10% chance of developing type 1. And then there's other percent chances of different autoimmune disorders as well. So in the context of type 1, that kind of tells us, well, presumably these beta cells, if they're producing an autoantibody, there's some level of self-activation going on there of autoinflammation, but for some reason they're able to withstand that, and it doesn't tip over to a threshold of actually developing to type 1 diabetes. And then in the context of type 2, actually, we see, depending on the study, anywhere a range from 25 to 35% of individuals with obesity can actually maintain that up to old age and actually never develop type 2 diabetes. So in many cases, it does turn over to type 2, but there's also observations that we can see people that maintain that, and presumably they're still having some level of inflammation going on because of this metabolic dysregulation. So altogether, I think that these kind of observations point to the fact that inflammation alone is not going to tell us the whole answer. There's something else that's going on differently within the beta cell compartment and how they respond, which is the main target of inflammation in these contexts in diabetes. So that's what I'm interested in. And I'm looking at that in particular in terms of how beta cells use glucose, how they metabolize glucose. So just to give you a little bit more information on that, Beta cells are really specialized kind of little machines that use glucose in a very different way than most other tissues in your body. And that makes sense, of course, because you would want, which they respond to glucose in order to secrete insulin, and you would only want that if your glucose levels correspond to when you're in a feeding state. So you don't want spurious insulin release when you're fasting, which would further lower and cause hypoglycemia, of course. And so beta cells have adapted the way that they handle glucose in order to really cause this ability of them to sense glucose differentially. One of the things is they have expression of a different hexokinase called glucokinase, and that has unique properties, which I'll go over in a little bit. And the other is we've pieced together a number of biochemists over the years have really been focused on this question over the decades, actually. And we've kind of pieced together using molecular studies and metabolomics that glucose really drives for mitochondrial handling of pyruvate in the TCA cycle. And this is important for producing coupling factors that are directly required as a metabolic signal to help push for insulin release. And apart from this context of insulin secretion, this happens within minutes. So you can take, beta cells or islets in vitro and stimulate them with high glucose, and you'll see them release insulin within minutes of that glucose being there, or seconds, actually. And so this is largely where we've studied this in the context of insulin secretion. However, over the past few years, we've also identified that glucose is not only important for the function, but it actually also ties into the fate of beta cells, whether they're going to be persistent and survive different stressors, or it also helps influence their ability to differentiate and maintain their identity, which we see going wrong in diabetes situations where you get dedifferentiation. And a lot of that work was started by Memo Achille. We also see an importance for glucose in proliferation, both acutely and chronically. And this has been looked at by many groups, in particular, the Weirs and Laura Alonso, who also used to be here at UMass studying this question. But in this context of these other processes, when it comes to the actual routes that glucose takes, we have a very little understanding in comparison to our deep knowledge of what it looks like in terms of insulin secretion. So presumably it's going to be different as well, because these things can take course over the period of a much longer time point, whereas insulin secretion happens within minutes up to a couple hours. And so the aspect of this that I'm interested in studying is the role that glucose has, or the routes that glucose takes in when it talks to pancreatic beta cell survival. And in order to look at this in a more deep molecular sense, I've actually targeted this enzyme glucokinase, which is the first enzyme that phosphorylates glucose and then starts its later breakdown of products. So this enzyme, and I've looked at this, of course, in the context of inflammation. So this enzyme is unique from the other hexokinase family members in that it has a really high Km for glucose. So that means when glucose is high, corresponding to your feeding levels, that's when it will be active. But if glucose is low, below a certain threshold level, it's not going to be active. So you can see in comparison to something like in neurons, you would want that going on all the time. You would always want glucose to be utilized. But in this case, when you have this glucose utilization coupled to insulin secretion, you don't want that happening. So this is really the reason why beta cells are glucose responsive in this sense. And then secondly, the enzyme is actually not product inhibited. So like the other hexokinases, it's going to have kind of a homeostatic role in the cell of low, constant glucose flux. By being product inhibited, it's not going to utilize glucose too readily. But in this case, as long as beta cells see high glucose, so as long as you've eaten your meal, and depending on how much carb is in there, if it's prolonged, your beta cells will maintain their activity and they'll keep secreting insulin. And then nature has taught us a lot about the importance. Oh, I'm sorry, I have this animation should have come up before. That's just the enzyme's name. But nature's taught us a lot about the importance of this enzyme in humans, because gain of function mutations are responsible for monogenic forms of diabetes. And these can be really severe and happen immediately upon birth. And then loss of function mutations are also really severe and can cause persistent hypoglycemia, which has a number of really adverse effects as well. So you can see from these mutations in humans that this tells us there's a really strong influence of glucokinase, the enzyme itself. So those mutations are in the enzyme on insulin levels. And so this was, a really attractive therapeutic target for many years. And there was a lot of excitement around it. But you could also see how it's kind of tricky because you wouldn't want to get too much activation or too little activation. And this is actually the same, this is mirrored by overall what glucose metabolism in general does as well. So what I mean by that is glucose metabolism can have different products that, if we think about it as a mitogen, it can produce beneficial species that will be supporting biosynthetic processes, cofactors, and really helping cells grow and supporting growth and survival. But we know glucose can also produce damaging products. And this really depends on the extent and duration of glucose exposure. For example, in hyperglycemia with diabetes, we see these toxic products actually being produced, and they come directly from glucose. So this question of looking at glucose in terms of it having beneficial or damaging effects, when I started my postdoc, it actually hadn't been looked at kind of like in an open, unbiased manner. People had done a lot of work with candidate approaches, like focused studies, but it hadn't been looked at kind of in a broad sense. And it hadn't been looked at in the context of inflammation. And so that's, and because of these pleiotropic effects of glucose, of course, we'd want to see in a more broad sense, everything that's going on when glucose is beneficial versus damaging. So I saw this opportunity to use tools that had been developed in my postdoc lab that were glucokinase activators, but they work differently than all of the commercially available allosteric activators that we had known about to date. And these commercial allosteric activators I had known about a few years before, because they were pulled out of clinical trials early because they had such potent effects on activating glucokinase. And I'll show you a little bit more about how the enzyme gets activated by them. But they had such potent effects that they were getting these damaging products being created and having secondary consequences. And so the clinical trials were stopped. But I knew about those and that I could use this kind of as a system to compare, because the activator that we had developed in the lab actually bound to a new site on glucokinase enzyme, which was near the active site for glucose. And it allowed it to maintain its glucose sensing properties, but still increased its activity. And this actually has protective effects. So in taking this system, I could ask the question, okay, let's activate glucokinase and then do so in one sense that we know has beneficial and effects on the beta cells and in terms of their function and survival, and compare that directly to a setting where we know there's damaging effects from other toxic products being produced. And then underneath that, uncover all of the metabolites and overall how the changes associate with either protective or damaging effects on beta cells. So just a little more information about the tools that we use. This is actually the proapoptotic member of the BCL2 family, BAD, the protein BAD. But we identified in my postdoc lab a number of years ago that when it's phosphorylated at serine 155 in the mouse or 118 in humans, you actually get a completely new and different role from apoptosis. So this blocks its role in apoptosis. It blocks its ability to bind the BCL2 family members and lower threshold for apoptotic activation. And it plays this positive role in metabolism through glucokinase activation. So we identified through structure function studies the specific residues on the BH3 domain of BAD. So these BCL family proteins have BH3 domains, and we identified the specific residues where it engages with GK. We're able to make mutants as well, a control we call the AAA that cannot interact and engage with GK, and it does not enhance its activity. So this is a nice control to have to look at specifically the GK activating abilities of BAD, and we remove this blocking of apoptosis that it also may play a role in the effects that we see. So looking at enzymatic activity, you can see in green is the phospho-bad mimetic. So we can either add on an aspartate mutation, or we can actually add to the peptide that's synthesized, we can add a phosphate group directly. And we see that with those, there's actually, if we look at the enzymatic activity of glucokinase here, you can see there's an increase in the Vmax. So this overall is higher than control, which is in gray here. And importantly, that the response to glucose, so as we increase glucose concentrations, it's kind of similar to the native enzyme. And then this important control, the AAA, when we look and compare to a vehicle DMS, which is a DMSO control, there's not much difference in terms of how the enzyme behaves in that context. And this work was done by Ben a number of years ago. So I took these tools and then looked at them now in the context of pancreatic beta cells, because we know beta cells express glucokinase. And so I wanted to verify that this was in fact the target for glucokinase, that it was on target effects on this enzyme. And we can take these peptides and biotinylate them and then do pull down assays. And you can see that GK is only coming down here from beta cell lysate with the phospho-bad mimetic, and with the AAA, you don't see anything. And then I used another BH3 control and you don't see GK coming out. And then I was looking towards structural studies in a deeper understanding of the integration so that we could get, better targeted small molecules made. So I did some isothermal calorimetry, and you can see the actual affinity for the ligand to the enzyme here is in the nanomolar range. Whereas, if we use the control peptide or a stapled peptide, it's hard to tell by eye, but it's significantly different. It's logs order less affinity. So that was pretty nice. And then took these, put them into human islets, and we can see the GK activity in this case is increased again with the phosphomimetic, or from now on, I'll just refer to this phospho-bad mimetic as a protective glucose metabolism. So with protective glucose on board, we get this increase in Vmax of the enzyme. And then if we compare this to the allosteric activators, you see not only an increase in Vmax with those, but they're actually left shifted. So now they're responding to glucose at much lower concentrations. And this is something that's probably tied into the reason why it has damaging effects, because we don't want these cells to respond to glucose at 1 or 2 millimolar, which is what it's doing. And I think this mode of differential activation of the enzyme is also tied into the effects that it has on beta cell survival. So what we can do, and I should also mention, when we work with pancreatic islets, we have a challenge in that there's multiple cell types there. So to get around that, we use different strategies of staining the beta cells only. And here I'm showing you some flow cytometry where we can look specifically at the beta cells and their survival rates by doing a co-staining. And you can see in response to a cocktail of cytokines, and this is a model that I use to mimic what the inflammation is like really in the type 1 setting, you can see that control islets treated with vehicle have a certain amount of death within 24 hours of seeing these cytokines. And then when we add on the allosteric activator, there's really not much difference. But when we add on the active site activator, the protective activator, you get now this huge resilience in these beta cells, and their survival rates go right up, even in this context of inflammation stress. And so this is kind of the system I set up to study. Okay, now that we see there's protective or non-protective effects of glucose, what are all the metabolites changed downstream of that? What is happening in terms of their metabolisms? So in order to do that, I set up an experiment, again, directly in the human islets this time. And we take the isolated islets from human pancreas from a number of different sources that we have, and treat them with either control PBS or cytokines, these are the cytokines that we use, TNF, interferon, and IL-1 beta. And those are relevant in the setting of type 1 diabetes. And then just take those and add on either the non-protective glucose or the protective glucose, and then performed a couple of different analyses, one that was kind of a broad catch-all approach, but also confirmed and followed that up with one that is more quantitative, actual chromatography-based mass spec approach. And so those data, and this is the information from the donors. So it's, we don't really get to choose the donors that we use, but so we try to, do equal representation of male and female, but unfortunately, if for any of you that work in this, there's usually a lot more male donors that we receive. And over the years following up, we would try to, recapitulate this in more of a broad sampling of donors as well. So what do these data look like? If we take them and plot them on a PCA, you can see that the different groups of treatments are really actually nicely grouping together such that even with donor differences, what we see is an overriding effect, first of all, from cytokines. So if you have cytokines on board, they're going to metabolically look very different than if there's no stress or no cytokines. And then also really differences between the two different ways of activating glucose. And then you can see that also in this heat map, but it's also just a massive list of metabolites. So we need to find a way to make sense of that. And for that, we run deeper statistical analyses and then pull out pathway enrichment scores. And of both the comparisons between islets that are just dying that are control versus protective metabolism on board, and then the additional comparison of protective glucose versus non-protective glucose, the top pathways that came out were actually involved in nitrogen metabolism and arginine metabolism. And that actually points to the urea cycle. That's where they're connected. But this was kind of weird because nobody thinks of the urea cycle when you think of beta cells. That's something that happens in liver or kidney, right? So for a while, I ignored it for a pretty good while and started following up on other candidates, wasted my time, and then realized, oh, wait, maybe I shouldn't have ignored that. So I started following up on that a little bit. And to do that, I looked at steady state changes in the metabolites within the pathway, which is actually pretty nice because if you look at the urea cycle, in general, arginase gets hydrolyzed and it forms this end product. The nice thing about that is in metabolism studies, we have this caveat of not knowing how much is going into production of a given intermediate versus its consumption. But in this case, an end product kind of tells us the level of activity going through a pathway. So I wanted to look at that. And you can see at steady state levels, just looking at urea, we see in human donors an increase with protective glucose metabolism on board versus the allosteric or non-protective activation really doing nothing to urea levels. The other nice thing about looking at arginine metabolism is we also know it can be oxidized by nitric oxide synthase, which is an important immune mediator. And we know that this produces nitric oxide in the case of inflammation is actually in part contributing to how beta cells die due to inflammation toxicity. So when I looked at nitric oxide levels at steady state again, I could see there's actually this reciprocal decrease in nitric oxide levels. So it looked like if urea is increased, nitric oxide at the same time was increased with protective glucose on board. And I wanted to test that whether these were on target effects and dependent on glucose metabolism. So in order to do that, I targeted the enzyme that they should be working through, which is glucokinase. So if we knock down glucokinase, I found that we no longer get this increase in urea levels, and we no longer see this retention in nitric oxide accumulation, but they shoot back up to kind of damaging levels, and the cells are stressed out again. So this is overall just steady state levels. And like I mentioned in metabolomic studies, we always want to have direct evidence of, it's considered kind of the flux of how much activity is going into a reaction. So in order to do that, I designed a flux experiment analysis where we use a tracer that's heavy labeled in the guanidino group here on arginine. And then we could actually track in the same cells, feeding this tracer to them, we could track the hydrolysis of arginine to forming two heavy labels in the urea group and the coproduct ornithine. And we could also simultaneously measure its oxidation through NOS and find a heavy labeled in the citrulline, M+1. And so when I actually gave this to human islets, I found not only was the overall levels increased, but the actual synthesis of urea was increased with protective glucose metabolism on board. And again, reciprocally, the generation of nitric oxide levels from arginine, so this direct contribution to this oxidative pathway, was also decreased with protective glucose on board. So this is really nice. I was seeing this kind of correlation If we have protective glucose, we get increased urea and decreased NO. But at the end of the day, it's still just a correlation, and we always want to have a causation kind of approach. And so to do that, I targeted the enzyme that makes urea to ask the question, well, I'm seeing this go up with protective glucose, but is it actually involved? Does it actually play a role in the protective effects? And so if I block this pharmacologically or genetically, and here I'm just showing you data from taking islets out of the whole body arginase 2 knockout mouse, that if you no longer have the enzyme that produces this urea present, you don't get these protective effects anymore. And I saw similar results with the arginase inhibitor. So one potential question is, okay, well, is the product actually protective? And that was really quickly and easily answered by, just treating cells with urea itself. If we just circumvent this, could we restore protective effects of glucose? And turns out that's not the case. So there was something more behind it. There was something more about the way that glucose is being utilized, the way that it's pushing arginine to be utilized. And so I took a step back at this point and thought about everything in the greater context. Okay, I'm targeting a glucose-dependent pathway. It's carbon-based. It's an essential carbon-based pathway in our cells. Somehow it's talking to a nitrogen-based pathway. How are they possibly connected? Well, if you go back to your Leninger Principles of Biochemistry 101, there's actually a theoretical link that hasn't really been directly shown, but it's a theoretical link through what would be the Aspartate-Argininosuccinate shunt. So you can get this amino acid being produced that can lead to, from using glucose carbons, that can actually go into feeding and influencing the urea cycle. So I did a couple of things, looked at steady state levels of aspartate, found that they were actually increased overall by protective glucose when you activate GK in a protective way. And then I found as well that if we target these enzymes pharmacologically or genetically, here I'm showing you genetic knockdown of these two enzymes, there's two isoforms, that you actually lose that increase in urea levels that you see. So that the protective effects working through to ureagenesis, it definitely requires aspartate in this context, and that suggests an actual direct link from glucose to the urea cycle. And more importantly, when we look at survival, so the protection that we normally get from enhanced protective glucose metabolism is actually lost if we knock down these enzymes as well. So this is a little bit indirect, so I wanted to go a step further and actually get direct biochemical evidence for this link being intact, and used a glucose tracing strategy. So this time, instead of the nitrogen being labeled, I'm labeling carbon in the glucose, and can follow that all the way into producing aspartate up to the urea cycle intermediate. And very relievingly, we actually saw something that confirmed all the other data to date at this point, was that the label was actually going all the way into the argininosuccinate shunt and the urea cycle. So that was very reassuring. And but the next question was, okay, well, aspartate coming from glucose actually has many different sources. There's cytosolic sources, there's mitochondrial, and as we know in beta cells, or actually in all cells, glucose influences many different metabolic pathways. So I wanted to use these glucose tracing strategies and take a deeper look at the actual source for aspartate. And to do this, I looked at the specific pattern of isotopic labeling. So when we take a glucose molecule and we label all the carbons, and then we feed this to beta cells or islets in this case, we can then track, okay, half of that is going to a pyruvate molecule, and then follow it along as it goes into the TCA cycle or stays cytosolic in glycolysis. And what I found of all the pathways that I looked at, I'm not going to show you all the negative data, I'm just showing you the positive one here, but only pyruvate carboxylase was actually increased by protective glucose metabolism. And this we can measure as an M+3 labeling of the intermediate malate. So this will actually not come from any other source of pattern labeling. And this enzyme, pyruvate carboxylase, has a known role in anaplerotic feeding into the TCA cycles. But this was the first time that it's been looked at kind of in the context of responding to inflammation stress in a protective way. So I wanted to assess whether it was directly involved and had a causal relationship here. And so I targeted it with a pharmacologic and genetic approaches. Here I'm showing you the genetic tools where we knock down PC or pyruvate carboxylase, and we actually get a loss in that increased aspartate that we see with protective glucose on board. And furthermore, we see a decrease in the ureagenesis. So it's actually involved, pyruvate carboxylase is actually involved in ureagenesis. And ultimately, what we care about most is it's important for the survival of these beta cells. So the normal protection that we see in this context of inflammation stress is actually lost if you knock down pyruvate carboxylase. But if you reintroduce an shRNA resistant PC, you now get restored protection. And then I saw similar things actually with the pharmacologic inhibitor phenylacetic acid as well. Okay, so just to summarize up to now, all of these kind of in vitro studies that I've shown have pointed to overall, I think, direct evidence that glucose can actually modulate nitric oxide levels, which that might actually have relevance in other physiologies where nitric oxide is important. It might be worth taking a look at. And then as well that PC has a ureagenic role, and this may help to kind of provide a direct biochemical link for clinical observations that have been made, 50 years back, and looking at people with PC deficiencies and that they have changes in their urea levels and lactate as well. And then importantly, I just want to note as well that pyruvate carboxylase and the enzyme in the urea cycle arginase that utilizes or hydrolyzes the arginine, both of those in and of themselves are necessary and sufficient for protection. So it seems like they're working within the same pathway to provide protection from inflammation stress. Okay, so at this point, everything I've talked about has been really in vitro context. But of course, we always want to know, is this going to actually matter in any kind of in vivo setting? And so to do that, I turned to using the diabetic mouse model with marginal mass islet transplantation. And we call it marginal mass because, so we put these, in this case, using human donor islets into the kidney capsule of diabetic mice. And it's marginal mass because if we follow their blood glucose levels for the whole experiment, they're going to maintain hyperglycemia. It's not doing anything for reversing diabetes in these mice. But it provides a nice opportunity to test with this suboptimal level, if we actually pre-treat them with our compound that we think improves their function or their survival, is that going to actually lower the blood glucose levels and have an influence on the same suboptimal number of islets being grafted? And so what we can see, if we look at the actual data, because this is just a cartoon to kind of describe what we're looking at. So in black is the control islets, then transplanted into mice. So they're treated first and then put into mice. And over the course of the experiment, these mice, this is their blood glucose levels, they maintain high diabetic levels. If we put in the allosteric activator, the non-protective form of activating glucokinase, we don't really see any effect. In some of the mice, it actually exacerbated their hyperglycemia. And that's why there's also large error bars, because it wasn't in all of the mice. But the case is really different if we add on the protective activator of glucokinase, the mimetic here. And we can see an early on immediate lowering of blood glucose levels in these mice, which actually persists throughout the whole experiment. And then if we get rid of these grafts by performing nephrectomy, you can see that the reversal of diabetes here was actually due to the exogenous human islets that we put in. And another way to look at that is that we measured human C-peptide and human insulin and found in the mice that it was actually the one that was increased. Importantly, if we block pyruvate carboxylase, we no longer see these protective effects against diabetes, and the mice are maintaining their hyperglycemia. And again, if we use a version of this phosphomimic that can't activate GK, we also see the mice are still diabetic throughout the experiment. So these effects are really GK dependent. But you might be asking the question of how is it possible that from what I've told you, that I'm pre-treating these islets and then putting them into mice, and up to 40, 50 days later, still seeing that there's a difference between just by having pre-treating them. Well, as it turns out, the answer is kind of early on. If we look at a time point where initial graft survival is really dampened by hypoxic stress, and immune attack or inflammation stress. In this case, and so I took a cohort of mice and stopped the experiment early and then pulled out their grafts and then looked what was going on and did some staining for beta cell death. You can see in the control graphs, there's a huge amount of beta cell death going on in the allosteric activator. That's also the case. But in the case of the phosphomimetic, there's some level of death going on, but it was nearly halved compared to the other conditions. So the amount of survival here is pretty much doubled in vivo when you're putting these islets in. And again, if we blocked the pyruvate carboxylase with this pharmacologic inhibitor, we no longer saw this reduction in beta cell death, and beta cell death levels just shot back up. And in the control peptide that doesn't activate GK, we also see a high amount of beta cell death in the blue here. So this was really nice, but another thing that occurred to me is, well, we're using, this is a xenotransplantation model. So we use mice that don't have an immune system. And I actually looked in these graphs and asked the question, like, is inflammation changing here? Is there an influence on that? But I couldn't find any infiltrates in the graphs. And I asked several other labs as well in the same kind of experimental context if they've ever seen immune infiltrates, and no one had. So this pointed to an additional protective feature going on beyond just inflammation responses. So then I went back to the metabolomics data and I looked at this and thought, well, what do we have in the graphs? We have hypoxia, oxidative stress going on. Is there anything that could point to that and how it would help against it? And it was really obvious, glaringly obvious, was glutathione was changed not far along after. So the higher, the more significant something is, which arginine metabolism and urea cycles at the top. And then more to the right was how much of the pathway is actually represented in those data. And so glutathione was at the top, and I wanted to target that next and used a pharmacologic approach, but also a genetic approach to do so. And in this context, I wanted to actually bypass the effects that we see of protective glucose lowering nitric oxide levels, which we know that happens through ureagenesis. So instead of just doing that, I wanted to apply direct nitrosative stress. So forget about its ability to lower nitrosative stress. If we add on oxidative stress directly and nitrosative stress, is there going to be a difference? And as it turns out, that's when I saw, oh, okay, well, this protective aspect of glucose is still protective if you add direct nitrosative stress. And then if we block the ability of these cells to produce glutathione with butadiene sulfoxide or with genetic knockdown of GCL, the enzyme that forms this tripeptide glutathione, we no longer see these protective effects. And so I wanted to dig a little deeper in this context and identify whether there was a direct link between the carbons that are promoted by glucose metabolism in this protective sense to go towards glutathione. So to do that, I used a different set of series of tracing analyses where we look at, and this also was done in human islets, where we can actually feed these islets labeled glucose and then track, throughout the different amino acids that are going to be generated. And there's actually a number of amino acids that are fed or fueled by glucose as well in these cells. And then look at the specific patterning. And what I found was that, first of all, if we just look at overall labeling, so if we just take everything that's labeled up here together and sum it all up, we can see that overall there's a significant increase in glutathione production and synthesis and labeling from the glucose carbons when you add on protective glucose metabolism. If you don't activate GK with this control peptide, you don't see those effects. So this is likely telling us, okay, it's dependent on glucose metabolism, and we see the carbons directly going from glucose to glutathione. So it's directly involved in its production. And then narrowing in on the specific pathway and the route that it might be taking has really pointed to a particular amino acid route that would form the M+2 isotopomers of glutathione. So we can see that in particular, that's the one that's increased in this context. And this gives us more information about the pathway it's taking and, can potentially identify new targets to enhance glutathione synthesis, which we know many cells like having more glutathione around. And then in this case as well, I wanted to look at what we know that pyruvate carboxylase activity is increased by protective glucose. So I wanted to see if it was also involved in this context. And I actually found that if you knock down PC, you no longer get this component of glutathione being produced from glucose. Okay, so just to sum up kind of everything at this point of what I've told you, I've identified that glucose in a protective setting can actually enhance pyruvate carboxylase activity. And this is a mitochondrial enzyme. This helps increase ureagenesis, which in turn lowers nitric oxide production and helps to fight against inflammation. At the same time, also through pyruvate carboxylase, you get increased de novo synthesis of glutathione. And this ultimately protects against oxidative and nitrosative stress in beta cells. And this, importantly, is independent of the known role that pyruvate carboxylase, which is mostly what it's been looked at in identifying its role in pyruvate cycles, and that increases NADPH, so which is also an antioxidant, and this is independent from the NADPH role that it has. And actually, these pyruvate cycles were actually also a nice example of how, so all this deep metabolism that we look at is in pancreatic beta cells, but in some cases, these pathways are actually relevant outside of the beta cell world, where, for example, pyruvate cycling was identified first in beta cells, and now it's just exploded of its importance in different cancers that are metabolically, sensitive cancers that have really taken advantage of pyruvate cycling. So that's something that we first identified in beta cells. Altogether, these pathways kind of reveal the umbrella effects that glucose can have in a protective sense, and ultimately how they influence pancreatic beta cell death. So those are some of the protective pathways that I've worked on. And now as I move into, so that was in my postdoc work, what I've talked to you about today. And as I'm moving into starting up my own independent lab, what I've been looking at, one thing I want to look at is the whole other side of the picture that I didn't talk about at all. So this was all the protective aspects of glucose. But I also found a number of really interesting pathways that are coming out of the non-protective side of glucose, and they likely will go along with what we see in hyperglycemia with diabetes. And so some of these pathways, for example, cholesterol, what I see when I label in human islets, a huge amount of cholesterol being produced from glucose. But when I look at mouse islets, there's almost nothing. It's pretty much zero. So that's an interesting, unique difference. Another difference, between human and mouse metabolism in the pancreatic beta cell. So that's one aspect I want to follow up on a little more. But what I'm most passionate about, I think, right now, and what I'll talk to you in a little bit more detail in the last couple slides, is this idea that, okay, so these pathways in beta cells are protective to the beta cell. But is it possible that those intrinsic pathways in beta cells have any influence on the metabolic environment? And could that also influence immune cell activation locally? So that's the next question. And these ideas are kind of, there's a lot of tools and reagents we have, and knowledge actually, that we can borrow from kind of the tumor microenvironment field that's going to really help, grow and expand these projects. So just, I want to mention as well, the immune cells that I'm looking at to start with. So we know that in the context of type 1, this has been studied in great detail in the adaptive immune side of things. But I'm actually going to take a look at something that's a little more broad and perhaps happening a little earlier on, as which is occurring in the macrophages. So in this context, I'm going to start with looking at the macrophages particularly, and that's for a couple of reasons. So this is a schematic of all of the important immune players that are present in type 1 diabetes. So of course, I'm paying homage to that and keeping it up here for now, but I am focusing on the macrophage for several reasons. They're actually the first immune cell that's present in the islet niche, and they're there from birth, so early on in development. And they're also, at any given time point, when in the absence of insulitis, they're the most abundant immune cell that's going to be present in an islet, in a healthy islet even. And there's recent evidence, so just a couple of things that we know about them so far, that they're important in initiating inflammation in type 2 diabetes in the context of obesity. And, presumably they're also involved in initiating inflammation in type 1 as perhaps antigen presenting cells. They should be firing probably early on in order to get the adaptive response. They're also taking on, there's some really interesting things that we've noted about them so far. So they take on a resident, more M1-like phenotype, which, you know, is the in vitro classifications of macrophages, but it refers to a more pro-inflammatory phenotype. And that's actually required for normal beta cell functioning. I think that might be because they're sort of in a barrier area that, you know, would be kind of like gut or lung macrophages where you need to have an environmental response mounted. So that could be what's maintaining their more pro-inflammatory behavior or phenotype. They're also involved in normal beta cell function and physiology. So if you deplete them by different means, genetically or pharmacologically, deplete the macrophages, I mean, you don't get proper beta cell mass growth or development, and you don't have normal beta cell function. And so far, all we know about them is that they are sensing beta cells. It's thought that every beta cell in an islet is actually in contact with a macrophage, even though there's only a few macrophages for, you know, hundreds of beta cells. But they're sensing beta cells. They take up insulin granules. They may actually supply IGF to beta cells in the context of type 2 diabetes. And in other contexts, acute inflammation, they are found to express purinergic receptors. So they may be sensing changes in ATP levels from beta cells. So these are kind of the knowns that we have about them. And the way that I'm thinking about this, I'll just show you the schematic, is that looking at the, you know, islet, the beta cell intrinsic metabolism could influence the metabolism outside the cells, and that this would then be taken up differently by macrophages and lead to either more pro or anti-inflammatory activation. And I think actually the field of immunometabolism itself is kind of, you know, provides a nice feasibility for the fact that changing the metabolite environment is actually going to influence macrophages to be pro or anti-inflammatory. And there's a number of pathways, for example, phagocytic activity, ROS generation, and cytokine production actually all require specific routes of glycolysis that we've worked out. And on the other hand, we know, I think my pointer's frozen, oops. We know that in the context of anti-inflammatory activation, this really requires PI3, things like AKT, mitochondrial biogenesis, oxidative phosphorylation, mitochondrial oxidation going on. So there's a lot of feasibility here in that just changing metabolism is going to change the ability of these immune cells to be active. And some of those pathways are actually what I'm going to be looking at first. So in terms of the pathways that I've identified from protective glucose metabolism, such as nitric oxide and then the Urea Cycle activity or arginine hydrolysis, we can see these play differential roles also in pro or anti-inflammatory activation. And so far what I've done is I've set up an in vitro setting to test this in. So I take human islets or mouse islets cultured in their media and then add on different changing their metabolic intrinsic features with protective or non-protective pathways, and then take that media, split it into two. So the same media goes for metabolite analysis and profiling, and then the other portion of it goes on to macrophages in culturing them and then checking if their activation is changed. And another way that I've looked at this is actual direct co-culture of the two and asking if we manipulate just the beta cells intrinsically, is that going to change the activation of these macrophages in order to kill them? So we pre-label beta cells or islets here, and then add them, co-culture the macrophages, activate the macrophages, and then see release of this fluorescence in a plate reader assay. And so some of the preliminary data that I'm getting so far are actually really, really reassuring, because you always want to know, you know, if you're doing something that's actually going to work, it's nice to know early on. So I have preliminary data that shows that these questions and this line of hypothesis is actually going to provide some findings here. So in looking at metabolites that have been changed, funny enough, one of the top ones that's changed if you add on protective glucose is actually arginine metabolite, and it's depleted in the protective sense. And then if we look at how this influences macrophage activation, we actually see anti-inflammatory markers changing, but also some changes in pro-inflammatory markers. So I'm still working out the details here, and I'll probably do some single-cell RNA-seq studies on the macrophages to try to get a better sense of whether there's population shifts within that. And then now if I take this a step further and look at arginine metabolism directly by using the knockout mouse system, we can see, you know, in black here is just macrophages co-cultured with islets with no priming done. So they're not going to attack beta cells without any kind of initial priming, or to a very low extent they will. And then if you prime them with interferon to be pro-inflammatory, you get this massive bump. If you knock out arginine, arginase 2, the enzyme that will use arginine, you get a sensitization. So there's even more killing of these beta cells. And that is actually dependent on the enzyme. So if we re-express it, you now have a restoration to normal killing levels. So the next steps I have planned for this are, you know, going to be moving into deeper kind of tests of, can we actually manipulate this dialogue now and engineer it between beta cells and macrophages and influence their outcomes on diabetes. So I have more in vitro studies to look at the actual routes that glucose is taking. We can trace and label, like feed the label to the islet cells, and then co-culture them with macrophages and see how or what is being transferred to the macrophages, how they're actually directly communicating with each other. There's a number of other molecular studies that I have planned to look at this, and then some in vivo models where we can actually test this communication directly, and in situ models as well. And then open that up to high throughput screening, looking at chemical libraries, small molecules maybe, and different gene sets to see if we can find, in a more broad, unbiased sense, kind of anything and everything that might be involved in this communication. And then I'm also going to take this to, you know, try to recapitulate what we see here in the mouse cells to humans as well. So we have plans, I have plans to look at that in human tissues that we get from different sources. Okay, so from all of that, there's a lot of people to thank over the years. I've trained some great undergraduates, and Lindsay, Nina, and Laura in particular contributed to some of the work I showed you today. And of course, my former mentor, Anika Daniel, our collaborators, Adolfo, who helped with showing us how to do transplantation studies, and Ivana Mills, who's showed me how to get macrophages, obtain them, isolate them, culture them, everything about macrophages and their preferences. And then Tatsuya and James for providing well over 100 human islets for our studies over the years, which is really not trivial at all. So that's a huge thank you to them. Edriggers, who has General Metabolics. This is a company that we did the non-targeted with, whereas all the targeted stuff we did in-house. This was kind of a broad, untargeted help with the untargeted stuff. And then our funding, of course, GDF, NIH, the Sternlicht Fund, and Harvard Stem Cell Institute QBRI research grant as well. Thank you.